Welcome to the section on insect vectors and alternative host plants of Aster Yellows Phytoplasma in South Africa. I'm Kerstin Krüger. Basic information on Aster Yellows Phytoplasma in South Africa has been provided in this program in the first training section on History of Detection and Symptoms in South Africa. This section is concerned with insect vectors and alternative host plant species. Phytoplasmas are phloem-restricted, wall-less bacteria that are pathogenic to many plant species worldwide. They have two hosts, plant host and insect host species, and rely on both for their survival. They spread through vegetative propagation of infected plant material, but mostly through the phloem-feeding insect vector species. Insect vectors of phytoplasma belong to leafhoppers, planthoppers, or psyllids. Phytoplasmas reproduce not only in their plant hosts, but also in their insect host, which retain the pathogen for life once infected. To identify insect vectors and alternative host plants, grapevine and the vegetation within and around vineyards and the natural vegetation in the fynbos and succulent karoo were sampled in the three regions where Aster Yellows Phytoplasma has been reported, that is Vredendal, Robertson and Waboomsrivier. We used different sampling methods for collecting insects. These include visual inspection, sweep netting, vacuum collection and yellow sticky traps. Insect samples were labeled and preserved in absolute ethanol for identification. At the same time, we sampled plant species growing within and the vicinity of vineyards and in the natural vegetation. The methods we used were described in more detail in this training program under Method of Sampling and Collection. The insects and plant species were identified by specialists or with molecular identification methods. In a previous study, we collected and tested more than 1,000 individual plants from 14 families and 33 species. The plants were collected in grapevine blocks and surrounding areas. Of the plant species tested, 11 species belonging to 7 families tested positive for Aster Yellows Phytoplasma. The table shows the plant species and the number of plants tested and how many were positive. Those that tested positive for Aster Yellows are highlighted in yellow. The positive species included a number of weed species such as blackjack and white goosewood and nettle. Further examples of alternative plant hosts are triticale, a cover crop in vineyards, and maize, here in the bottom image interplanted with young grapevine plants in a grapevine block. Mgenia fuscovaria, the vector of Aster Yellows, transmitted Aster Yellows to triticale and maize in the laboratory, and this species has been observed feeding on a number of weeds in vineyards, like on nettle as shown in the image at the top. Although we currently do not know whether Mgenia fuscovaria is able to transmit the phytoplasma from these plants to grapevine, the number of Aster Yellows positive plant species suggests that weed management is important to reduce Aster Yellows sources. In addition to the survey in vineyards, we surveyed alternative host plants for Aster Yellows in the fynbos and succulent karoo biomes in the 3 regions where Aster Yellows occurs. The survey resulted in only 2 Aster Yellows positive plant samples out of 989 individual plant samples from 19 families and 42 species. The 2 Aster Yellows positive samples were from Mesembryanthum crystallinum. This plant species is native to South Africa. It is widely distributed and also occurs in other parts of Africa, Australasia, Asia, and Americas and Europe. The low number of Aster Yellows positive plants suggests that the plants within vineyards and surrounding areas are more likely to serve as Aster Yellows sources than plants in the natural vegetation. After the discovery of aster yellows in South Africa, a partnership between industry and research organizations led to the identification of the leafhopper vector Mgenia fuscovaria. The leafhopper is indigenous to South Africa, but little is known about its distribution. It has been recorded in the winter rainfall region in the Western Cape province and in the summer rainfall region in KwaZulu-Natal and Limpopo provinces. The species feeds on shrubs and trees. Adult Mgenia fuscovaria are fairly large and robust leafhoppers which are approximately 4 to 6 mm long. They can be recognized by the shape of their head and the arrangement of the large compound eyes. Mature adults can be recognized by their brownish color and the whitish spot in the center where the forewings join, as well as by the black insides of the hind legs. The nymphs of Mgenia fuscovaria are pale to bright green and resemble adults. The wings develop gradually. Young adults are greenish and turn gradually brown. The seasonal abundance of Mgenia fuscovaria in Vredendal in the Western Cape has been monitored since 2009 with yellow sticky traps. The dark blue bars in the graph on the top show the mean abundance, that is, the mean number of leafhoppers caught between 2009 and 2016, and the red line shows the number of leafhoppers collected following a prolonged drought from January 2017 to December 2019 in the Western Cape. The abundance of Mgenia fuscovaria declined dramatically from 2017 following the prolonged drought, suggesting that the risk of aster yellow spread during this time is low. The three peaks in February, summer, June, winter and September suggest that this species has 3 generations per year. Vredendal falls into the winter rainfall region in South Africa, which is characterized by a Mediterranean climate. The bottom graph shows that most of the rainfall is received during the austral autumn and winter from April to August. Temperatures are highest from November to February. In general, Mgenia fuscovaria numbers are highest during June, when the rainfall is highest. The lowest numbers were caught during November and December, which together with January and February are the hottest months in the region. The following is recommended for the management of aster yellows phytoplasma in grapevine based on our current knowledge. Sanitation measures to prevent disease spread is very important. This includes the planting of only certified aster yellows free plant material, marking and removal of infected plant material such as shoots and roguing of infected plants. Mgenia fuscovaria is active all year round. Nymphs and adults have been recorded on grapevine during the growing season and on weeds, some that are hosts of aster yellows during the remainder of the year. The recommended strategy is therefore to monitor the leafhopper not only through the growing season but throughout the year with yellow sticky traps. Management decisions and timing should be based on the presence and population size of Mgenia fuscovaria, vine phenology and climate. Weed management is essential as weeds from different plant families are hosts of aster yellows phytoplasma and may serve as the phytoplasma source. In addition, planting of crops that are hosts of aster yellows, such as maize, between young grapevine plants should be avoided. Insecticides for foliar application or soil drenches that have been registered for the management of the insect vector should be used based on the presence and population size of the vector according to the manufacturer's specifications. The different control measures have been included in a management plan for aster yellows. The management plan can be found on the TROPICSAFE internet site under reports. This brings me to the end, and I would like to acknowledge the following persons and organizations for their support. Thank you very much.